I'm Matthew Hall at Bomley Emergency Courses, and in this short video tutorial I want to talk about adaptive immunity, and in particular about the humoral and cellular immune responses. Adaptive immunity is the immune response to a specific pathogen or pathogenic antigen. And for the purpose of these tutorials I've broken that down into 4 steps or stages. In a previous tutorial I talked about antigen expression on cell surfaces in association with major histocompatibility complex molecules, MHC molecules, and the recognition of those antigen MHC complexes by clones of T cells via their T cell receptors. We also discussed T cell clonal expansion, where clonal T cells, activated by binding to antigen MHC complexes, go on to proliferate and differentiate into mature T cell types, helper T cells, cytotoxic T cells, and memory T cells. So in this tutorial we take up the story where we have activated or armed helper T cells, which now need to go on and trigger the effector responses of adaptive immunity, and that is the humoral or antibody mediated response and the cellular or T cell mediated response. So let's start the story with the humoral immune response. Now the humoral immune response is about B cells, plasma cells and antibodies or immunoglobulins. And the first thing to realize is that initiation of a humoral immune response requires the activation of antigen-specific B cells. In general, this requires 3 separate signals. First of all, the B cell must bind directly to its target antigen via a surface B cell receptor. Be clear that no MHC proteins or molecules are required at this stage, and the B cell receptor binds directly to its target antigen, usually an antigen on the surface of an extracellular pathogen, perhaps a bacterium. As well as binding target antigens, B cells also require stimulation from cytokines, which in turn have been released from activated or armed helper T cells that we were talking about in the last tutorial. And in addition, there may be further co-stimulatory signals required from direct receptor-receptor interaction with those helper T cells, those armed helper T cells. Once activated, B cells go on to proliferate and differentiate into antibody-secreting plasma cells. And it is important to note that the antibody secreted by plasma cells has an identical affinity for the same antigen that activated its precursor B cell. We'll talk more about antibodies and immunoglobulins in the next tutorial, but for now, let's recognize that there are 5 classes of antibody: IgA, IgD, IgE, IgG, and IgM, and each of those has a different tissue location and a different role within the humoral immune response. The primary function of antibodies, however, is to bind target antigen on the cell surface of pathogens and promote the removal of the pathogen by activating either complement-mediated cell killing or macrophage-mediated cell killing. So antibodies don't kill pathogens themselves, but they coat the pathogen by binding to pathogenic antigens and then promote removal or killing of the pathogen via activating complement and macrophage-mediated killing. Some activated B cells differentiate into memory B cells, and these memory B cells provide a faster and greater antibody response on future re-exposure to the same antigen or pathogen, and in so doing contributes towards immunological memory. As a general rule, most extracellular infections, extracellular pathogens, are cleared in this way by the humoral immune response. Here is a summary diagram of that. We have an antigen-presenting cell digesting a bacterium, breaking it down into its constituent antigenic fragments and expressing those fragments on the surface in association with MHC class II molecules as an antigen MHC class II complex. Helper T cells recognize that antigen MHC class II complex via their T cell receptors and become activated or armed. B cells here in turn become activated through firstly binding their target antigen on the cell surface of a pathogen, for example, via their unique B cell receptors. The B cell also needs to receive co-stimulatory signals from the activated or armed helper T cell, either through cytokine release or direct receptor-receptor interactions, or more commonly, both. Activated B cells go on to proliferate and differentiate into plasma cells, and it is the plasma cells that secrete antibody into the circulation. Antibody bind their target antigen, and note it is the same antigen that activated the helper T cell and that also activated the B cell precursor. And the antibody bound to pathogen goes on to activate either complement or macrophage mediated killing. A small number of activated B cells go on to form long-lasting memory B cells, which reside in the lymph nodes long after the infection has been cleared and contribute towards immunological memory. So now let's go on and talk about cellular or T-cell mediated immune response. The cellular immune response is the T-cell response to body cells that have become infected with intracellular pathogens. And to be clear, the cellular immune response does not involve antibodies at any stage, which clearly differentiates it from the humoral response. And the way it works is that cytotoxic T cells become stimulated by cytokines released from active or armed helper T cells. Those active cytotoxic T cells are able to recognize infected host cells by binding to their cell surface antigen MHC class I molecules, and those antigen MHC class I complexes mark host cells as infected and therefore targets for cytotoxic T cell killing. The cytotoxic T cells attach to and kill the infected cells by releasing cytolytic proteins called perforins, and these proteins insert into the target cell membrane and effect cell lysis. And as a general rule, most intracellular pathogen or infection is eradicated by cytotoxic T cells in this cellular response. And here is the cellular immune response summarized in a diagram. Here, a typical body cell that has become infected with an intracellular microbe, perhaps a mycobacterium or a virus or a parasite. Protein shed by the microbe within the cell is picked up by the cell and expressed on the cell surface in association with MHC class I molecules as a antigen MHC class I complex. That complex is recognized by the T cell receptors of helper T cells. The helper T cell becomes activated through that interaction and releases cytokines. Those cytokines stimulate a population of cytotoxic T cells, which go on to lyse infected cells through recognition of the same antigen MHC class I complex on the cell surface, which activated the original helper T cell back here at the beginning. Killing of target cells is brought about by the release of perforins from the bound cytotoxic T cells, and those perforins are protein channels which, when inserted into the target cell membrane, effect cell lysis and killing. So that's the summary of the cellular immune response, which is primarily responsible for clearing intracellular infections. I hope you found that useful, and I have one more tutorial in this series which goes into antibodies and immunoglobulins in more detail.